Howard, talk to us about adoptive T cell therapy and where that may fit in.
Yeah.
What's that science about?
So really the use, so we've already mentioned the central role of T cells, and so part of the thinking around adoptive cell therapy is that if the T cells are being suppressed in the patient with a cancer, maybe you could take those T cells out of the patient and really kind of re-engineer them to be much stronger at either recognizing or mediating effector functions against cancer. And so there's interestingly been a lot of approaches to this. So one possibility is you could actually go to the tumor site where the tumor reactive T cells might be, and even though they may not be as effective as you want them to be to clear a tumor, by taking them out of the body, separating them out, and then exposing them to other growth factors such as interleukin-2, you could grow them up, make them stronger, and then give them back to the patient. And it turns out that that actually works pretty well, especially if you can pre-treat the patient sometimes with non-myeloablative chemotherapy to get rid of the existing T cells. Some people have used irradiation to do the same thing. And really, this has been a lot of the pioneering work in the surgery branch, which has shown a lot of success there. Another strategy is to genetically engineer these T cells so that if they're not recognizing the cancer, you could put in potentially a T cell receptor that targets a known tumor antigen and use those. And more recently, there's been work using what's called chimeric antigen receptor T cells, or CAR T cells for short. And these use almost an antibody chain that can target an antigen, and then you can actually tack onto those various T cell stimulatory domains, and then you can transfer those back into patients. And that's shown particular promise in CD19 expressing hematologic malignancies, where that work is actually far along.
So, John, and actually I'd be interested in Howard's opinion on this too. Is every cancer immunogenic?
Yeah, it's a great question, and Howard brought up something that I think is a very important idea that people are thinking about, but it also points out the complexity. And it's the following idea that if you've got a lot of mutations, you should have a lot of new epitopes, and you should be more immunogenic. Well, we know some cancers that have very high mutation burdens, but are not particularly immunogenic. Small cell lung cancer is one, where even though it's very genomically unstable and you've got, the chromosomes are just a mess, but the immune system doesn't recognize it that well. So it suggests that there are mechanisms that the tumor has to suppress the immune system even when it does have a high mutation burden. Pancreatic cancer has a fair number of mutations. It doesn't tend to be immunogenic. So this illustrates that the number of potential epitopes isn't the only predictor of whether something is going to be immunogenic, and we have to start understanding all the different components of what suppresses the immune system and what can stimulate it. The other thing is that things that are immunogenic are not always mutations. There's a whole family of antigens out there, related to, we call these the testes antigens. And so the MAGE A3 being an example, MAGE A10, there's a whole family like this. And they're not mutated, but they're typically suppressed during the normal adult development, where they may come on during certain parts. So the immune system still recognizes them as foreign. And so these are some of the new approaches that are being taken, is can we use that as something to stimulate the immune response? Because tumors often express these.
Yeah.
I agree. I think there are different ways to get to immunogenicity, whatever that is. So if you don't have a good antigen, we now know that there may be other factors such as radiation, local radiation. We've been hearing about this abscopal effect where you radiate a tumor and you can see regression elsewhere. We think that's because you're generating potentially new antigens at the site of the radiation, which can then stimulate the immune response. The other interesting story that's emerging is that in addition to this mutation burden, some cancers that are mediated by a virus, for example, HPV tumors, the polyoma virus and Merkel cell carcinoma, may also stimulate immune responses and make these tumors immunogenic in a way that is not dependent on the mutation burden and neoantigens. So that may be a completely different mechanism, but will make those tumors immunogenic.
Yeah. That's an interesting thought, because if you've got an antigen that the tumor doesn't depend on and that isn't obligatory, well, you may mount an immune response, but the antigen may just get turned off, and that's how you could escape. But if you've got, for example, an HPV positive head and neck or cervical cancer, every cancer cell's got HPV, and I haven't ever heard a mechanism described to sort of get rid of the virus there.